Hi, this is the Probing Crowded Cells with Deep Learning Group, and our team consists of Flora, me, and Neel. And so today we would like to talk a bit about the projects that we've been working with in CS Plus this summer. So to begin with, structural biology is a field in which biologists are trying to determine the structure and thus function of macromolecules such as proteins and enzymes inside of your body. By doing this, we're able to better understand how they actually function and thus develop targeted therapeutic approaches to, say, target a functional site or to alter the structure in a way so that it doesn't function in the way that it might. As such, there are many different computational and experimental ways to determine. One exciting new development in this field is AlphaFold, which is able to predict enzyme structures and enzyme and protein structures directly from the genetic code. And this strides beyond its competitors, and it's very exciting. So since we do have a theoretical approach, now what exactly is our experimental approaches? So first of all, there is Cryo-EM and NMR, and these are traditional approaches which use isolated and very purified samples of the target protein or target macromolecule in order to determine their structure, either through imaging or through their chemical components. As such, these are very limited in the scope that they remove the macromolecule from its native environment, thus isolating it from any interactions it may be having with its environment or other macromolecules. By doing this, we lose an aspect of the functionality that could be obtained from the samples. And as such, there has been a development of a bridge between imaging macromolecules on the structural scale and between imaging it as a whole that is called Cryo-ET. In Cryo-ET, we take a slice of a cell or another biological sample and then basically use the electron microscope to create something called a tilt series. Which is a bunch of images at different angles. By creating this tilt series, we're able to get a sort of 3D representation of the sample, and we're able to use the tilt series to reconstruct the sample into a 3D tomogram. So as you can see here with number 2. And the most important step comes next, which is to actually find out the structure of the macromolecule we're interested in. So in order to do this, we need to find many copies of the macromolecule inside these scans, because as you can see in the image, the particles themselves are very small and low resolution. This is a result of the limitations of the electron microscope and the fact that cryo-ET scans often result in very low signal to noise ratio. By finding every single particle inside of the tomogram and averaging it, we're able to generate a high definition representation of the figure. And as such, our project is mainly focused on the second step, where we're trying to pick the particles from the tomograms. So this has already been shown to be very useful in Cryo-EM scans, which is one of the earlier traditional methods that we talked about, where the sample is highly purified and not necessarily interacting with anything besides itself. As such, there are certain object detection approaches that have been used in these scans to successfully particle pick. And we want to apply these to Cryo-ET scans, which currently are either manually picked or picked using template matching, which is limited in that it's very computationally expensive, and there's only so many templates that you can choose from. So certain macromolecules may not be able to be identified using template matching.
Our pipeline for particle detection is divided into 2 stages. In the training stage, we first obtain a dataset of tomograms. We then manually annotate particles and background noise in the tomograms and extract their 3D coordinates as the ground truth positive and negative samples. With those ground truth coordinates, we generate training data and feed them into a neural network classifier to get optimal weights. In the inference stage, we first run a sliding window through the whole tomogram and use the classifier to get a window's probability of being positive. With this list of coordinates and their probabilities, we use non-max suppression to generate coordinates of predicted particles. When we generate training samples, we have two approaches. In the 3D approach, we simply extract a 20 by 20 by 20 or 10 by 10 by 10 cubic volume centered in the particle coordinate from the tomogram. However, since the 3D tomogram is reconstructed from 2D tilt series, there are some reconstruction artifacts due to the missing wedge and limited tilt angles when we take the scan. The goal bits, which are not the particles we want to detect, are usually classified as the particles by our network. We couldn't really filter them out as they don't overlap with the annotated goal bits due to the reconstruction artifacts. Thus, we also propose a 2D approach. In this approach, we directly map the 3D coordinate of the particle onto its corresponding location on the tilt series. Then we extract an area in each tilt image to generate a projection stack for the particle. We also try to average the projection stack to boost the signals. Our classifier is implemented using 3D ResNet since it has useful packages and is effective for image classification. The architecture is shown in the figure. We choose ResNet because its skip connections can help prevent vanishing gradient and make the network much deeper for the classification tasks. In the inference stage, we use a common approach of sliding window. Basically, we extract a cubic volume from the tomogram in each possible location and obtain its probability of being positive with the classifier. This figure shows how sliding window works in a 2D image. Then we use non-max suppression to suppress all the locations that have low probability of being positive and get rid of overlapped windows. To calculate the overlap between windows, we use the common IOU metric, which is the intersection over union for two windows, as illustrated in our slides. Different IOU metrics represents different levels of overlap.
So this diagram outlines the output of each stage of our data pipeline. First, we start with the tomogram, and then using our trained model, we perform binary classification to generate a probability map with predictions for each coordinate, which is in the middle. And then on the right, we have the non-max suppression output, and non-max suppression will process the clusters of high probability predictions to produce the individual particle coordinates. Then from there, we can overlay our network's predictions onto the tomogram and view the results. So on the left, you can see one layer of the tomogram, and you can see the particles in it, which are those little black dots. And then on the right, you can see the results of our best performing 3D approach, which is the model trained on 3D subtomograms. And in this picture, we've color coded the results. So green circles are particles that are true positives, meaning that they were classified correctly as positive. Red circles are false positives, meaning that they were classified incorrectly as positive. And then we have blue circles, which are false negatives. And those are basically the ground truth coordinates that we didn't identify. And the results you can see on the right are pretty encouraging because the model you're looking at right now is only using 10% of our training data, which means it performs well without a lot of ground truth labeling required. These are some statistics that we use to gauge the performance of our network. Along the x-axis, we have some of the data transformations we tried to improve performance. And along the y-axis, we have F1 score, which is an object detection metric that basically uses the number of true positives, false positives, and false negatives to generate a single number that we can use to judge performance. And as you can see in the chart, the radius 5 models significantly outperform the radius 10 models. And in this case, the model that was not normalized and used a Gaussian filter, which is just a blurring filter that removes noise in between the particles, performed the best. So we use the same process with non-max suppression as the 2D approach to generate the predictions for the 2D approach as well. And then we can overlay the results for each approach and compare them like you see here. And one thing you'll notice is that the 3D approach outperforms the 2D approach pretty significantly. You can see that there's a lot more green true positives in the 3D approach and a lot more blue false negatives in the 2D approach. One thing that is encouraging about the 2D approach, though, is that the way it handles gold bead artifacts. And gold bead artifacts are the projections of gold beads in the tomogram surrounding the actual location of the gold bead. And those are caused by the missing wedge effect from when we reconstruct the tomogram from the tilt series. And so we hypothesized at the beginning of our project that since the 2D approach does detection on the tilt series, it basically skips the tomogram reconstruction, that would do better at handling these artifacts. And you can see in these pictures that it does. The 3D approach identifies the gold bead artifacts as false positives, whereas the 2D approach does not. Yeah, so ultimately this was a pretty challenging problem for us. One of our goals was to use as little training data as possible, since the whole point of using machine learning to improve particle detection is to use as little human labor for manual annotation as possible. And so that made it difficult to improve performance. But also using non-max suppression and the sliding volume gave us a lot of different hyperparameters to experiment with and tune. And so that made it difficult to improve performance as well. And although the 2D approach did not perform as well as we would have liked it to, the fact that it managed the gold bead artifacts so well is promising for the technique and is definitely worth further research.
So to end this talk, we'd love to give a big thanks to our mentors Shang-Fu and Wendy, as well as our PI, Dr. Bartasagi, and the CS Plus program. Thank you very much, and we would love to answer any questions or hear any comments.